Hey, welcome back, Casibang's crew. This is the brain part 2. We've gone over all of the parts of the brain and the different things that the brain, the different functions that the brain has. And now there's just a few things, sort of miscellaneous things that we're going to put together that we want to talk about in terms of what the brain does, or different things that the brain does for us. So, our objectives for this lesson. We are, again, identifying key contributions of researchers into the development of tools for examining the brain. And then we're going to discuss the role of neuroplasticity in traumatic brain injury. Okay, so first of all, how did we learn all of this information? How do we know what these parts of the brain do, where they're located, all of that information? So the first thing that we're going to talk about is having our heads examined. Okay, so what tools do scientists and doctors use to help us learn about the brain? The very first tool that was ever used is lesioning. Right? And when you lesion the brain, you are actually cutting into it. So, way back when, this was the only way that they were able to study the brain. And so obviously this was done after people were dead. And they're cutting into the brain and potentially damaging parts of the brain. So we learned a little bit through lesioning, but obviously there are a lot of methods around today that are a lot better. The first one is the electroencephalogram, EEG for short. And yes, you do need to know the shortened versions of all of these that I'm going to give you. So this is a picture right here of an EEG. Okay, so you're hooked up with these electrodes on your brain, and it's going to a computer, and it's just kind of giving you these brain wave activities. So to show different electrical activity that's happening in your brain. And you might be hooked up to this while you're sleeping to see your brainwave activity. You might be asked to read something or to talk or to look at colors or look at pictures, any of those kind of things, so that you can see the electrical activity that's happening in your brain. The next one is the CT scan. It's computed tomography. And this is basically a type of X-ray for your brain. And again, here, we're really looking at the structure of your brain. We're looking at taking a picture of your brain to make sure, or to compare it to other images of other people's brains, just to make sure that the size is correct, there's no tumor growing or anything like that. So basically a picture so that we can see the structure of your brain. The PET scan, positron emission tomography, is where you are injected with radioactive glucose, and then pictures are taken of your brain. And I'm gonna show you some images of that in just a second. But the radioactive glucose actually lights up different areas of your brain. And again, you may be asked to read or to have a conversation or look at some pictures. And they're taking pictures of your brain to see what areas of your brain light up with this radioactive glucose. An MRI, magnetic resonance imaging. So this one uses magnetic fields and radio waves to, again, map out the parts of your brain. And this image right here is showing you an MRI, and it is kind of showing you some of the images that you can get. And again, we're looking mostly for the structure of your brain to make sure that there's no damage or nothing growing that shouldn't be there or anything like that. And then the last one, and this is the latest invented, is the fMRI, which is a functional MRI. So here we're looking at our brain functioning as well as the structure of the brain. So the first brain imaging that looks at both the function and the structure of the brain. Okay, so this is an image of an MRI, and you're looking at this arrow right here. We're looking at this part of the brain right here. So this image here, and you notice the difference, okay? So this is a normal, healthy brain, and no, I'm sorry. This is the brain that has schizophrenia, and this is a normal, healthy brain. So here's where, again, we're kind of looking at the structure. You notice difference here. You notice some difference here in the folds. Okay, so when you notice this, you can see that there are some brain differences that happen when you have schizophrenia. Here is the image of the PET scan. So the PET scan here, and like I said, you're lighting up certain areas of your brain. So here's a normal brain. Okay, and what we're looking for are the red and yellow and orange parts of the brain. Those are the areas that we're looking to light up. So the middle brain here shows someone with a very mild cognitive impairment, okay, which just means they might be intellectually a little bit lower on the intelligence scale. And then here's a picture of someone that has Alzheimer's disease. So again, you see the really big differences in the brain here. Alzheimer's, this middle section, doesn't look like there's anything there, like they're kind of missing part of their brain. And there's a lot less of the red and the orange and the yellows of a very highly active brain. Moving on to the brain's plasticity. So brain damage is obviously a really huge issue, but there have been a lot of studies done on whether the brain can regenerate itself. Can you come back from these types of brain injuries? I'm sure you've heard people say, you know, oh, you're killing brain cells. Okay, so if you kill your brain cells, will they come back? That's a big thing. So plasticity is our brain's ability to change and adapt when things go wrong or when things happen. So when we talk about plasticity, this image here is a picture of Gabby Giffords. She was a senator, and she was shot in the head. So obviously she had a lot of brain damage. She couldn't speak for a long time. She couldn't walk for a long time. It was a miracle she really survived. But after years and years and years, she has been able to relearn how to walk. She has been able to relearn how to talk. So here's where we see this amazing ability for our brain to be able to regenerate even under really, really gross damage. And then neurogenesis. Neurogenesis happens when our brain develops new neurons in our brain. And obviously neurogenesis happens a ton when we are a fetus, when we are growing in mom's belly. It's all about neurogenesis. We're building all of those hundreds of billions of neurons in our brain. But this process of neurogenesis does continue throughout our life, just not as much or on a grand scale as it does when we are fetus. So in the last video, we talked a little bit about how we have a left hemisphere and a right hemisphere, okay? So the two different hemispheres of our brain, and then right down this line would be our corpus callosum, which is that network of nerve fibers that connect our left and our right hemispheres together. So I did kind of briefly describe to you that there is brain lateralization that happens, which just means that our left hemisphere does certain things and our right hemisphere does other things. Okay, so for example, okay, in our vision, on the left hemisphere, we're looking at mostly words and letters, or we're processing words and letters. Whereas in our right hemisphere, we're processing patterns, faces, and emotional expressions. In terms of hearing, our left hemisphere is in charge of our language. And this is really, really important for you to remember. Okay, I always put the left or the L in left with the L in language. And then our right hemisphere is looking at non-language sounds like music. Okay, our memory, our left hemisphere is in charge of verbal memory, where our right hemisphere is in charge of non-verbal memory. And then language, our left hemisphere, speech, grammar rules, reading, writing, arithmetic, right? Most of the aspects of language, where our right hemisphere really focuses on the emotional tone of speech. And then our spatial ability in our right hemisphere, we're looking at geometry, our sense of direction, distance, mental rotation of shapes. All right, so what happens if that corpus callosum is damaged or is split for any reason? So Vogel and Bogen were researchers that really looked into this concept of the split brain. And what they did is they researched what the corpus callosum did for us as far as it makes sure that those messages are sent back and forth from the left hemisphere to the right hemisphere and vice versa. And then Myers and Gonzaga really looked at what would happen if we split that corpus callosum. And the reason that they did that is they did this procedure called the split brain procedure. And really what it was for patients that were suffering from epilepsy. And when you have epilepsy, you have seizures. And those seizures tend to start in your brain. And they thought, what if we split that corpus callosum and those messages don't go? So maybe if the epileptic seizure starts in the left-hand side of our brain, if the corpus callosum is split, then it doesn't go to the other side of our brain. And maybe we can stop some of these epileptic seizures. So they did the split brain procedure where they went in and they did brain surgery and they would sever a person's corpus callosum. And this is an actual image of a brain. And this is the corpus callosum section of it. So what would happen if you had that split brain procedure? So basically what happens is it is very, very effective for people with epileptic seizures. It does seem to stop or significantly decrease the seizures because, again, those messages can't go from one side to the other. And amazingly, doing this split brain surgery doesn't really have a major adverse effect on people. You can survive very happily, very healthily, and very cognitively. You can survive with this corpus callosum being split. But it does affect some small aspects of your life. So what I want you to do, just on a piece of paper or even on your video notes, is just to try this. So I want you to try with your left hand to draw a circle and your right hand to draw a square at the same time. Okay? At the same time, with your left hand, you're going to draw a circle, and your right hand, you're going to draw a square. So take a second and try and do this. It's really difficult, right? Most of you will probably not able to do it, but someone who has had this split brain surgery can do this. Okay? And they've done a lot of research with split brain patients to try and figure out why this is. Okay. So very quickly, I'm going to kind of try and go through this, and we're going to talk more about this in class. So what we know is that when we see something, I'm going to put the entire picture up here. When we see something in our left visual field, okay, so this is a pencil, it goes into our eyes and it's processed into our right hemisphere, okay? So here it is, it's processed into our right hemisphere. And then again, when we see something in our right visual field, like an apple, it's processed over here into our left hemisphere. So because things are processed into our opposite hemisphere of our brain, these patients with the split brain procedure can do things a little bit differently than people with complete brains. So this is a visual that kind of shows some of the experimentation that they did. So they have someone who has a split brain procedure. They're looking at a computer. They're told to look at the dot in the middle of the computer. And then a word is put. So we see the word heart, but it is split by the dot. because we know that the H and the E are going to be processed in our right hemisphere and the ART are going to be processed into our left hemisphere. Now, when we ask this person, what word do you see? They're going to say art, because that is in our left hemisphere where our language is, okay? Where our language ability is. But then if they're asked to point with their left hand to the word that they see, they are going to answer something a little bit differently, okay? So we're going to do a little bit more. You're going to watch a short video and we're going to do a demonstration on how this split brain procedure happens, or the differences in split brain patients, how that happens. All right. So it's all about knowing what each side of the brain can and cannot do, particularly with language and speech. So knowing the hemispheres, knowing that the hemisphere controls the opposite side of the body. So we're going to ask some questions. So if Sally is a split brain patient and sees a baseball in her right visual field, where does it go in her brain? Okay, it's going to go to her left hemisphere. So then, can she say she sees a baseball? Okay, can she say that she sees a baseball? Yes, because language is in our left hemisphere. Okay, and the image is in our left hemisphere, so she'll be able to tell us. If she holds a baseball in her left hand, can she say she's holding a baseball? Okay, why or why not? Okay, so no, she cannot say she's holding the baseball because the right brain received the info and it doesn't have the language ability. Okay, the right brain received the info because she's holding it in her left hand, and the right brain doesn't have the ability to speak. Okay, so that is it for this video. Please, please, please write down your questions, come into class with them tomorrow, and we will talk more about the split brain procedures in tomorrow's class. Bye, guys.